you. So hi everyone, my name is Shannon Pinnell, and like was just said, I'm currently a senior studying electrical engineering at NC State. And today I want to tell you about the worst day of my entire life. Well, maybe it wasn't actually the worst day of my entire life, but it really felt like it was in the moment. Let me explain. I'd worked in a few research labs throughout freshman and sophomore year of college. I helped build a robot that sorted tiny fossils to track climate change, and a biosensor that detected valproic acid, which is an epilepsy drug, in human blood to help doctors make better dosing decisions. From those experiences, I knew I loved the process of research, but I wasn't super passionate about the work that I was doing. So during the summer after my sophomore year, I joined a biophotonics, you had the pronunciation just right, lab working to create a watch that measures blood hemoglobin levels using light instead of the traditional blood draw. You see, the most common form of nutritional deficiency in preschool-aged children worldwide is iron deficiency anemia, or IDA, which has been linked to poor school performance, increased behavioral problems, and just kind of overall negative outcomes for children. The World Health Organization estimates that 40% of children under 5 have IDA. So this is a very impactful issue for lots of people. So what's the cure? It's simple. Consuming more iron, either in the form of iron-rich foods like tofu, beef, or beans, or taking an iron supplement will get you right back to where you need to be. So it sounds like a pretty easy fix. Why are so many people still struggling? Well, it's a diagnostic issue. For people with easy access to medical resources, an IDA diagnosis is pretty easy. It requires that your doctor order blood tests to see if your hemoglobin levels are within range. And hemoglobin is a protein found in red blood cells that allows oxygen to move from your lungs to other organs in your body. It's also responsible for taking the carbon dioxide byproduct of cellular respiration back to the lungs to get exhaled. And guess what your body needs to make hemoglobin? Iron. So the way an IDA diagnosis is supposed to work is you get your hemoglobin levels checked via blood draw, they're out of range, and you make changes to your diet or supplement routine to eliminate the IDA and you feel better. But think about all the things that go into a blood draw. First, you have to transport yourself to a medical center. Once there, doctors have to have access to the tools to do a safe blood draw. That means sterile office space, available needles, enough skilled medical professionals to actually do the draw, facilities to keep the blood cold while it is transported to a separate lab for processing. You have to have all the needed reagents and equipment to actually do the test. And once you have the output of the test, you have to communicate with the patient and family what the result is. So imagine you're one of the 400 million people the World Health Organization estimates to not have access to quality medical care. A blood draw for a child presenting with fatigue is just probably not going to happen. It's way too resource intensive. So how do we fix this? I believe the best option is to create a better way to diagnose IDA that does not require the resources of a blood draw. This is a project I've been working on for the past 2 years. We're using a method called continuous wave near-infrared spectroscopy to determine the amount of venous hemoglobin present in the wrist. And the way we do this is by shining 3 wavelengths of light through the tissue and then detecting how much light is present at 2 different distances on the wrist. And the trick is we shine the light at a very specific wavelength. Oxyhemoglobin, the hemoglobin with the oxygen, absorbs light a lot at about 830 nanometers, and deoxyhemoglobin, the hemoglobin without the oxygen, absorbs light at about 770 nanometers. And we are lucky that other things in the body don't super absorb light at these wavelengths, or at least it's enough that we can do some math tricks to cancel out their contribution. So if we shine a known amount of light through tissue, and some of the light gets absorbed by the hemoglobin, then we can put a light detector at the end of the tissue, and we would know how much light got absorbed while it was traveling. And then using some math, we can convert that light value to a hemoglobin value. Now, unfortunately, I don't have time to get into the math today, but we can totally talk about it later if you want. Essentially, we take the ratio of the light put into the tissue with the light detected at the end of the tissue, calculate the slope for a bunch of different data points, and use some known absorption coefficients to calculate our hemoglobin values. Sounds easy. We know exactly what math we have to do and what values we need. Now we just have to build it. So that's what I did. I spent a year picking out parts, designing circuits, putting those onto a circuit board designer and testing the circuits. On this slide, you see a flow chart of the system that we ended up designing. So the LED is told to turn on with the microcontroller, which is kind of like a little computer. The light travels through the tissue in this curved shape due to the scattering and refraction effects of the specific tissue we are putting it through. That light is then picked up by the detectors, which output a certain amount of voltage based on how much light they see, kind of like a solar panel. Now, the voltage signal that comes out of these detectors is super small. The tiniest amount of interference can completely change the value of the voltage that we are measuring, which is what we're using to calculate our hemoglobin values. So if you have an error in this voltage, we are totally throwing off our hemoglobin calculations. But voltages that are bigger are more resistant to interference. So we can amplify our voltage using this gain block to make sure our value isn't super affected as much. Once that is done, it goes to the data acquisition module, which samples the data and sends it to the microcontroller. And then the microcontroller sends it via Bluetooth to a smartphone for the doctor or whoever to look at. And so we built it. On this slide, you can see the LEDs outlined in red and the detectors in blue, but there were some issues with this design. That super small voltage I was talking about, in the short distance from the detector to the gain module, it was picking up interference just sitting in the lab. And that was super not okay. Our data was insanely not right. So we had to figure out where that interference was coming from and do something about it. And so we took lots of measurements in all sorts of different conditions. In the lab, outside, in perfectly dark rooms, everywhere we could think of. But the interference was basically the same the entire time. So we thought about our experimental design. We had changed every environmental variable except for what was already on the circuit board. So that got us thinking. Was it the other circuitry on the board that was causing the interference? So we went back and looked at our design, and sure enough, there was a wire right next to our output wire that was probably causing some interference. So we redesigned the board so that we had one big board and two little boards with our detectors that connected together. On this slide, you can see the design file on the left and the manufactured board on the right. Our hypothesis was that the extra plastic would insulate us more from other signals, and it had the added bonus of putting the LEDs more flush to the skin, so ambient light wasn't getting caught under and getting into our detectors. And we did testing and the interference was gone. Perfect. Now there's some other issues that we're still working on, but that was a huge hurdle. So now back to the worst day of my life. I was part of a program that funded my work that semester. It was awesome. They paid me to do research. And if any of you get the chance to do an REU, which is a research experience for undergraduates, I highly recommend it. Usually they happen over the summer, but this one was during the semester. Anyway, part of this program was that I had to give a presentation in front of faculty members and experts in the field that I was working in about my work. And when I tell you I was scared out of my mind for this talk, I mean it. I prepared for weeks, making sure I could talk about why my research was important. I made sure my diagrams were correct. I took pictures of my work and I reviewed how everything operated. And it came to the day of the talk and I gave my presentation and I told them everything that we had done and what our plans were moving forward. And I finished and I asked if there were any questions. Now I was the fourth student to go on this day, and each student only got about one question. Usually the professor just wanted to go back in their slides to see a graph again or to see a picture again. But I asked for questions and I saw 4 hands go up. 4. I was terrified. The first question hit me out of left field. They asked how hemoglobin levels change over the course of the day. Well, I had no idea. I had spent the last year looking at electronics, not blood. But I pulled myself together and said in a very polite way, I'm not sure. I will get back to you on that. 3 hands left to go. The second question wanted me to go back in my slides to graph. Easy, perfect, got it. 2 hands to go. The third person asked why we didn't use a specific type of part for one of the electronics. Well, the truth was I had never heard of that. So I said in what I hope was a way that sounded smart that I didn't know what they were talking about. That seemed to go okay. So 1 to go. Now this person was a big researcher. He was in charge of multiple labs. He gets tons of funding and is super well respected. He asked about a specific detail of the math that we were doing to get from the light levels to the hemoglobin levels. And the truth was, it had been a long time since I had looked at any of that math. I was trying to diagnose electronics issues. So I did what I could, and I told him the name of the paper that outlines the math, except it somehow didn't come out right. I told him to read a paper by Suzuki to know what the math was, not thinking that this person does research in this field. Of course he has read Suzuki. Everyone has read Suzuki. Essentially, what I had told this person who had dedicated their entire life to this field was that he needed to learn the basics of biophotonics. It was like telling Einstein to go read an algebra textbook. I was mortified, but sat down and listened to the rest of the presentations. Later that night, I got an email from my professor. He said, "You did a great job." Great job? What are you talking about? I made a fool of myself. A couple days later, I was talking to a mentor and she ended up laughing at me, which I thought was a bit odd. I didn't think the situation was funny at all. But eventually she stopped laughing and told me that I had the experience that many people will have when they start presenting their research. When you give a presentation to experts in the field, they're going to bring up questions you never dreamed of. So it's it's common to not know the answer. And the best thing to do is to admit you don't know and learn the answer for next time, and eventually it gets easier. Over time, I made a conscious effort to extend myself grace during my work. If I made a mistake, I tried to not let it get to me, to just note down what I could do better next time and move on. And my mentor was right, it does get easier. Later, someone asked me how hemoglobin levels changed throughout the day, and I knew the answer. There were also a lot of questions I didn't know the answer to, and that is okay. Because I know as I continue working in my field, I will gather knowledge and experience, and I trust myself to become an expert one day. And that is what I hope you come away with today. I hope you know that research is fun and rewarding and valuable, but I also hope you know that mistakes happen and you will probably say something dumb in a presentation, and that is okay because you are very early on in your research journey. And there's still so much time to learn and explore. To finish out, the other day, I saw the professor I told to read Suzuki, and he smiled and waved at me, and I waved back and continued down the hall, and everything was fine. So my advice to you is to try to let go of perfectionism and give yourself permission to make mistakes. That's the end of my talk. I'm happy to take any questions in the chat. Thank you.